Hello everyone. Thank you for joining us to this ISCB-SC webinars. I am Ashik Mahmood. I would like to remind you if you want to know about the upcoming webinars or how you can present your research and be part of this initiative, you can follow us on Twitter as @ISCB-BSC. Today we have an interesting talk by Mr. Mohak Sharda. His background has a background in bioinformatics and applied biotechnology. Currently, he is doing PhD in India from National Center of Biological Sciences. Today, he will present his scientific finding on evolutionary and comparative analysis of bacterial non-homologous end joining repair. So, thank you, Mr. Mohak Sarda, to be with us. Now, the floor is yours.
Okay, thank you so much, Mahmood. Hello, everyone. Happy New Year to all of you, first of all. I'm very excited to be presenting my work today at ISCB webinar series. I'll be talking about this work that I have been doing in my PhD on evolution of non-homologous end joining repair in bacteria. And we have used computational approaches to target this question. So DNA is important for the continuity of life across all life forms and across all organisms. And it is composed of these four nucleotides, A, T, G, and C, which form the DNA double helix. And we have a backbone for the DNA double helix, which is composed of sugar and phosphate moieties. Now, DNA is not inert, it is a chemical entity. Therefore, it is prone to a number of different kinds of insults. And one of the most lethal kinds of insults that this DNA can undergo is called as the double-strand break, which occurs when there is a break in the sugar phosphate backbone, when there is an attack on the sugar phosphate backbone. And a number of factors can cause double-strand breaks, like X-rays and gamma rays, which can cause chromosomal rearrangements, and in severe cases can also lead to cell death. So then it becomes imperative for all organisms to invest in mechanisms that can repair such double-strand breaks to the DNA. So with that, broadly speaking, organisms have two broad kinds of repair pathways for double-strand break damages. The first pathway is called as the recombination-based repair, or the RBR. Now, over here, whenever there is a double-strand break, the organism can use the intact second copy of the chromosome, the homologous copy, and can go ahead and repair the DNA in an error-free fashion. On the other hand, the other kind of repair pathway is called as the non-homologous end joining repair, also abbreviated as NHEJ, and that is what is going to be the topic of our talk today. And for this particular pathway, there is an unavailability of the second copy of the chromosome. So what happens is that the organism prioritizes stability over accuracy and can repair the DNA, and in doing so, it can introduce insertions, deletions, and so on. Now, what we know in eukaryotes is that this sort of a repair is controlled in a cell cycle controlled manner, where, for example, in the G1 phase, we know from studies that the recombination-based repair is actively inhibited in the G1 phase, and NHEJ is the one that can repair under an event of a double-strand break. But in the S phase and the G2 phase, when the organism has the second copy of the chromosome during or post-replication, then recombination-based repair takes a front seat and can carry out the repair. Now, as it follows, then in eukaryotes, both these pathways are evolutionarily conserved. But in prokaryotes, one can then draw an analogy and think in terms of the growth phase, growth curve. So in the log phase, when the nutrients are available, there is an active cell division and DNA replication, because of which recombination-based repair can be used in case there is a double-strand break. But in the stationary phase, in bacteria, which spend the most of their time in the wild in the stationary phase, recombination-based repair just cannot act. And so non-homologous end joining repair takes a front seat at this phase again. But surprisingly, what we know is that from studies that in prokaryotes, while recombination-based repair is evolutionarily conserved, NHEJ is not. And that was sort of fascinating to us, and what led to us as a motivation to ask the question that what does it mean for bacteria like Escherichia coli to lack NHEJ, whereas those like Mycobacterium tuberculosis to harbor it? And with that, we defined three objectives for our study. The first was, how is NHEJ distributed across bacteria? In the sense that, is there a particular pattern to the phylogeny where NHEJ is present or absent? Then we asked, what is its evolutionary history? So what processes could have led to this evolution of NHEJ? And then finally, the third question we asked was, what evolutionary forces could explain the presence or lack of NHEJ among bacteria. So before we move to these objectives, I would like to tell you briefly about what NHEJ in bacteria exists as. So whenever there is a double-strand break, the first protein that comes into play is called as Cu, which comes and binds these free ends at the double-strand break site. Then the next protein is recruited by Cu, which is called as a ligase D or ligD. And this ligase D is again composed of three domains: the polymerase domain, the nuclease domain, and the ligase domain. Now, if you remember from your chemistry classes, for the ligase to ligate the DNA, you require these two moieties, the 5' phosphate and the 3' hydroxyl group. So in case this moiety is not present at the damaged site, polymerase and nuclease, they do the end processing and make sure that these moieties are then repaired using the ligase, using an ATP to give an intact DNA back. So because of this, even in our study, we have called an organism as NHEJ proficient if they code for both Cu and ligD. So then first we asked was, how is NHEJ distributed across bacteria? So with that, what we found was that we could divide our organisms. So we took a data set of around 6,000 bacterial genomes, and we could divide them in 4 groups. The first group was the extreme right in red, that is the NHEJ negative organisms, which did not have either Cu or ligase D. Then the second kind in yellow is Cu only, where we could only find Cu but did not find ligase D. And then the third type in green were ligase D only with no Cu associated. And again, in this, we had 2 kinds, the non-conventional ligase D, which we have termed like that, where all the 3 domains were present in the same protein: the ligase domain, the polymerase domain, and the phosphoesterase domain. And any other combinations of these domains in the proteins were termed as non-conventional ligase D. And as it follows, then the 4th state that we defined was the NHEJ positive state, again corresponding to the kind of ligase D which was present. So one such example of conventional NHEJ that we know from experimental studies is Mycobacterium tuberculosis. And similarly, with respect to the non-conventional NHEJ, which lacks the phosphoesterase domain, is the Bacillus subtilis organism. So what we find is that in our data set of 6,000 organisms, 1/4 of these organisms have NHEJ, and the rest do not code for NHEJ, or they have an incomplete machinery. So then the next question we then asked was if there is a pattern to these 22% bacteria that code for NHEJ. So what we then did was we constructed a phylogenetic tree. What you're seeing over here is a phylogenetic tree, a species tree of some representative organisms out of our data set. And the names are color-coded on the basis of the clade to which they belong. So for example, the yellow clade that you can see on the top right is the proteobacterial clade. And what we find is that as compared to archaea, so archaea again is a protein involved in homologous recombination in bacteria, which is present throughout these species, the other 4 domains which are associated with NHEJ have a very patchy distribution across all of the phylogeny, thereby reiterating the fact that there is no pattern as such if you look at the whole phylogeny. So then we asked, what is the evolutionary history of NHEJ evolution? Then for that, what we did was we did something called as ancestral state reconstruction, where we start with the extant species on the extreme right. So in this example, the 3 extant species I'm talking about are ABC. So if I say X is the common ancestor of ABC, I'm referring to this particular node over here. So we tested against 3 models based on, so let's say if I'm testing only 2 states, NHEJ negative and NHEJ positive state. So the forward rate is rate 1 and the backward rate is rate 2. So what we did is that we changed the equation of these 2 rates. So in equal rates model, the 2 rates were allowed to be equal, and then they were sampled from their posterior distributions using the Bayesian approaches. And similarly, we tested for all these models. And then finally, we used something called as stochastic character mapping, where we could, at each of these nodes, starting from the extant species till the time we reach the root, we could draw from their posterior probabilities the most likely state at that particular node. And what we find is, again, this is a schematic. I'm just for the purpose of this talk, I have made a cartoon representation of our findings. You can look at the full figure in the paper. So what we find is that the eubacterial ancestor most likely was devoid of NHEJ. It was independently gained across multiple lineages throughout evolution. And having said that, it was not sufficient to stabilize the trait because there were multiple secondary losses and gains as well, therefore, as you reach towards the extant species. Now, to gain NHEJ, that can be thought of in 3 ways. Either you can gain NHEJ by gaining both the proteins at the same step, and one of the ways that comes to mind is through horizontal gene transfer. The other two ways again could be that you either gain Ku followed by ligase D, or you can first gain ligase D and followed by Ku. So what we find is that the most common kind of gain in our data set was the one where the ligase D was gained first during evolution, followed by Ku, giving a complete NHEJ machinery. And then again, in the reverse direction, one can again think of three scenarios on how to lose NHEJ. In our data set, we found that the most common kind of loss was a direct loss, where both the proteins were lost at the same step. Now, we also looked at the time spent in each of the states across the whole bacterial phylogeny. What we find is that the least amount of time that was spent was in the Ku-only state. And then we also looked at the number of transitions. So over here, you're seeing a state diagram where the arrows represent the transitions to and fro each of these states. And the width of the arrow represents the amount of transitions in a particular direction. So what you can see from this is that not just the time spent, but even the transitions to and fro Ku only state were rare. And this suggested that the presence of Ku alone in the genome might have been costly in the genomes. And from studies, we know that Ku has been known to interfere with recombination-based repair. So what it means is that if just Ku is present, it would not be able to carry out NHEJ, and it might even interfere recombination-based repair in certain conditions. Next, we then checked the impact of horizontal gene transfer, as I already discussed. So horizontal gene transfer, very briefly, is nothing but an exchange or possibility of exchange between two species which might not be closely related. So the arrow that you can see over here in the figure, in the horizontal arrow, represents one such horizontal gene transfer event. So the first observation that we saw was that in our dataset, 60 organisms belonging to different clades carried NHEJ on their plasmids, and we know that plasmids can be the hotspot for HGT. Then we went ahead and we checked even further if we could detect HGT in our dataset using different methods. So the first method that we looked at was a parametric method based on the differences in the genome, the base composition of the genomes. By this method, we could not detect any signal of HGT for NHEJ proteins. So then we turned towards phylogenetic methods, where what we saw was that by the way, for this analysis we also included archaea in our analysis. So we did the homology detection for archaea as well, and we again found that in our data set only 4% organisms had NHEJ. Rest of them only coded for Lig domain, and there was no archaea that had just the Ku and did not have the ligase domain. So we used two methods, the approximately unbiased and mental test correlation methods, to check for incongruence between gene and species tree. So if there is an incongruence between the gene and species tree, it might mean that it could have had horizontal gene transfers within the species under our analysis. So first thing that we found was that if you look at the gene tree, species tree incongruence, what we find is for RECA, which is our positive control, we could find a good amount of congruence between RECA's gene tree and the species tree, corresponding species tree. But it was not the case for Ku and ligase D. As you can see that the archaea, which are in black, and bacteria, which are in red, are mixed up in the Ku and the ligase D. So that suggested that it could be because of horizontal gene transfer. Then we asked the question if these were only pertaining to closely related species, or was it also the case that distantly related species also underwent HGT. So for that, we did a mental test of correlation. So on the x-axis is the correlation coefficient from 0 to 1. And as you can see that as compared to RECA, Ku and ligase D distance matrices had a very low correlation coefficient, suggesting that these were not limited to closely related organisms. And then we asked that incongruence between trees could mean that processes other than transfer, like duplication and losses, might also lead to incongruence. So then in that direction, we used a DTL model, the duplication transfer loss model, and we predicted donor and recipient cells under the transfer events. So what we found was that in this figure, in the left-hand side is the Ku transfers that you can see, and on the right-hand side are the ligase transfers across bacteria and archaea and within bacteria. So what we find is that we could detect transfers not just between bacteria and archaea, but also between or within the different bacterial phyla. And just to give you an example, if you focus on this asterisk on the left figure over here. This is the transfer event which is between archaea and the ancestor of firmicutes and actinobacteria. So then we took the Ku gene sequences. We did a principal component analysis of the multiple sequence alignment, and what we see is a beautiful cluster on the extreme left bottom, which has archaea in red, actinobacteria in blue, and firmicutes in green. So thereby again reiterating the fact that there could have been a horizontal gene transfer among these species. Finally, we looked at what evolutionary forces could explain the presence or lack of NHEJ among bacteria. And for that, we chose two central genome characteristics, which I will be talking to you one by one. So the motivation behind choosing them was that these two characteristics could determine the availability of the second copy of the chromosome. So I'll be coming to that in a moment. So first thing that we found was that if we compare the distribution of genome sizes across organisms that have NHEJ and the ones that lack it, we find that organisms that have NHEJ tend to have larger genome sizes. And we did two sorts of tests over here. One was a non-parametric Wilcoxon test, but we also then followed it with a test called as phylo-ANOVA, which also controls not just for the chance events that usually statistical tests do, but it also controls for phylogeny. So when you even apply the phylogenetic control, we still do see there is a significant difference between these two populations or these two sets of NHEJ proteins. Now, then the next thing one can ask is if this is just an artifact that probability of finding a gene in a larger genome would be higher as compared to a smaller genome. So to test that, if that is the case for our data set as well, what we did was we took a genome pool of 6,000 bacteria and we selected certain number of genes, so in our case 920 genes from this pool at random. And then we went back and we looked at the genome sizes for each of these genes that they came from. And we finally calculated the median genome size. And this we repeated for 100 iterations to get a random distribution of median genome sizes. And then we finally compared this random distribution that you see over here to the number of the genome size of organisms that have NHEJ, the median genome size. And what we find is that, in fact, the genome size of organisms that have NHEJ tend to be significantly greater than what one would expect at random. And then the next, so just to give you a motivation, just to think of how genome size could be associated with NHEJ repair. Now, I want you to imagine two genomes. One is a smaller genome and one is a larger genome. Now, let's say I tell you that they both start replicating at the same rate and at the same time. After T units, if I tell you that the smaller genome has replicated half of its genome, then the bigger genome would have 1/4 replicated its genome. And at this point, if there were double-strand breaks that happen at these four sites, then the probability of using the second copy of the chromosome and thereby recombination-based repair would be higher in a smaller genome, or would be more effective rather in the smaller genome. Thereby, one can imagine or one can expect a higher selection pressure to maintain NHEJ in larger genomes. And that again matches with what we saw in our data set as well. Second kind of characteristic that we took was growth rate. Now, for growth rate for our data set, we took a number of rRNA operons as a proxy. So studies have shown that there is a strong negative correlation between number of rRNA operons and generation time. So one can think of rRNA operons as being a part of the ribosomal machinery, which helps in translation. So more the number of rRNA operons, more would be the potential for a higher or a greater protein turnover. So I want you to just remember that for us, for this analysis, the more the number of rRNA operons, more would be the growth rate. So that is the proxy that we have taken. So what we find is in our data set that organisms that have NHEJ tend to be slow growing than organisms that do not have NHEJ. But when we apply the phylo-ANOVA test, which controls for phylogeny, we see that signal getting lost. And this distribution is broad for NHEJ negative organisms that you can see in orange over here. So this is broad, but then most of them are dense towards the higher growth rate side. And again, you can think of the same motivation in the sense that if now imagine you have two bacterial genomes of the same size this time, and they both start replicating at the same time, but different rates, then after time T units, again, you can imagine the one with the higher growth rate would effectively be able to repair the double-strand breaks. So thereby, one can expect a higher selection pressure of maintaining NHEJ in organisms that have slow growing or have a slower growth. Then we next ask, do changes in genome characteristics occur independently of NHEJ state, or are these changes more or less likely in NHEJ positive lineages? So is there a correlated evolution between the two genome characteristics? So for that, we compare two models, the independent model, where we had the two characteristics over here, NHEJ and genome size, and we allowed them to evolve along the phylogeny independent of each other, so with their associated rates, which are mentioned over here. And we compared it to another model that is a dependent model, where changes in one trait might affect the changes in the other trait. So that is the dependent model. And if you closely notice over here, both of these models are nested among each other. So when we use maximum likelihood and Bayesian approaches. Again, I'm not going into the details for the interest of time, but we see that there is a signal of correlated evolution in NHEJ and genome size. And just to visually reiterate that point, one of the strongest correlation evolution signals that we got was in this clade of actinobacteria. So on the top, you are seeing Mycobacterium genus, and on the bottom, you're seeing Corynebacterium. So as you can see, the ancestor of both these genus had NHEJ, where Corynebacteria lost it, Mycobacteria retained NHEJ. And then if you look at the ancestral reconstruction of genome sizes on the figure on the right, you see that when the NHEJ was lost, it also corresponded to a loss of or the decrease in the genome size as well. And then similarly, we also got signal for correlated evolution for growth rate and NHEJ. And visually, again, taking the same example with Corynebacteria that has lost NHEJ, there was a corresponding increase in the growth rate, which is the colder color. So colder color over here represents higher growth rate. And finally, we looked at the combined impact of genome characteristics on NHEJ state. We used phylogenetic logistic regression. Again, it is logistic regression, but it also controls for phylogeny. And for that, we used, so for our two independent variables were genome size and growth rate. Our binary dependent variable was NHEJ. We also tested for multicollinearity, and because we could not find any, we proceeded and we tested 3 models. And what we find is that genome size is the best predictor for NHEJ state. So in summary, what I have told you today is that NHEJ was found to have a sporadic distribution across the phylogeny. And they most likely in the ancestral state did not have NHEJ. It was gained multiple times throughout evolution, and also corresponding to the secondary losses and gains as well. We also found that a Ku state was the rarest, and then how we discussed how the state could be costly in the light of what experimental studies have also suggested. And then we also discussed how horizontal gene transfers could have played a major role in the propagation of NHEJ, and these transfers were not pertaining to only closely related bacteria, but even distantly related bacteria as well as archaea. And finally, we talked about 2 genome characteristics, and we discussed how genome size could be related with NHEJ presence with a larger genome size and a slower growth associated with NHEJ presence, and that these might be correlated, these might be related in a correlated fashion, and that the best predictor of NHEJ state was genome size. So with that, I would like to acknowledge my PI, Ashwin Sainer and Sesha Sai, and my co-PI and mentor, Anjana Badri Narayanan. I would also like to thank all my lab mates for being supportive throughout, and my funding agencies, NCBS, TFR, TDU, DBT, Government of India, and ISCB, not the last but not the least, ISCB, to really giving me an opportunity to present my work today. So with that, I would thank you all, and I would be happy to take any questions. Thanks.
It was a really wonderful presentation, Mr. Mohak.
Thank you.
Now we have time for questions from attendees. So
Right.
please feel free write in the chat box available on YouTube.
Sorry? Okay, should I read them?
Uh, no. Keep it open. So please
Okay.
feel free to write in the chat box available in YouTube for question. Any questions from attendees? In the meantime, I want to ask a question
Right.
to Mr. Mohak. Okay.
Yeah.
Uh, Actually, it was a really nice presentation, and everything is really clear.
Thank you.
So I want to know that how we can apply this knowledge in our day-to-day life.
Yeah, what knowledge? Sorry, can you please rephrase the
Uh,
question?
of your finding in our future study.
With respect to, okay, so one of the long arching things that can come out is that we have now described a pipeline or a way which can be extended to even other sort of repair pathways. And because we know that repair pathways can have a huge role on how the genomes evolve, right? So we can then start asking similar questions in terms of other repair pathways. For example, we are also in the lab looking at other repair pathways, not just NHEJ, and we are asking questions if we can see if there is a dependence on bacterial evolution and these repair pathways. So that's one of the long-term goals that can come out of the study.
Oh, nice.
Yeah,
I guess there is no questions.
no problem.
So I'd like to inform everyone that we will have another exciting webinar at 19th January. Everyone is welcome to join. So thank you, Mr. Mohok, again.
Thank you so much for hosting.
We'll, hope you will come again with your new scientific finding in future.
Definitely. Thank you so much.
Thank you, everyone. Also, so bye for today. Stay safe.